Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is meant for 10th October 2020, and we have started off, as I promised you, the practical side of it. So henceforth we will be going side by side. The theory part of it first will be covered. As such videos I have sent, we are doing the revision. So as and when we start revising a particular chapter, concerned or related practical part of it also we will cover, so that your understanding of the concept will be clear. And this is basic procedure how to do the practicals. And you can also keep one biology practical record and start writing this. And here I want to give you some clues as well as instructions. Basically, any experiment, when you are writing in the record, you start off with aim or object. So you start off with aim or object. Like you have got to study parts of compound microscope, its proper use and maintenance. So you are able to see now clearly. So this is the aim. Then second one is materials required. That is compound microscope, a permanent slide, a practical notebook, all that. And then once the requirements are over, you are going for description in such experiments. If you are involving any activity, then we say procedure to be followed. So here, whenever there is a procedure, you must write it in the past tense. Okay, don't write in the present tense, because you are doing it. Once you come back to school, individually you will be doing the experiment. So since you are doing it, you have to write point wise every sentence in the past tense. And once procedure is over, then you go for the observations. Based on the experiment that you have done, what is that you have observed? Since you are observing, so that also should be point wise and past tense. After observation, based on your observation, you conclude. Write the conclusion. Conclusion can be past tense, present tense, anything. I leave it to you. And then after the conclusion, you will write precautions. What are the precautions that you have taken while performing the experiment? So that also can be in present tense. So in past tense, mainly procedure, okay, observations and result. This you can write. And the main thing, two, procedure and observations, you must write point wise and in the past tense. This you must keep it. And in your record, on one side there will be lines, okay, wherein you will be writing like this, and on another side there is a plain page, white page. On the white page you will be drawing the diagram, okay, like so. With that instructions in mind, what is required now? You need one practical lab book wherein you start writing that. So as part of your syllabus, we have got experiment number 1 to study the parts of compound microscope. Of course, once you come back to the school, you will be touching it, operating it, feeling it, all that. At present, you can only see, and I will be sending one virtual labs, O-labs it's called, sent by CBSE. So you can log into that and you can see the virtual mode of doing the experiment also, as long as you are at home. So with that background, now I am going for this experiment number 1, study of the parts of compound microscope. So microscope is an instrument, okay, microscope is an instrument which is widely and extensively used in the biology laboratory. It is used to study the details of objects which are too small to be seen with the naked eye. The development of the microscope can be considered to be the most important factor which helped tremendously in the advancement of biological sciences. A microscope increases the size of retinal image, the image formed on the retina of the eye of an object. The ratio of this increased image to that of the formed on retina of an unaided normal eye is termed as magnification of the microscope. The ability of a system microscope to distinguish two close points as two separate points is called resolving power of that system. Human eye can discriminate objects which are larger than 100 millimeter. It means the resolving power of human eye is 100 microns or 0.5 micron. The resolving power of compound microscope is 0.3 microns. Thus, the compound microscope not only increases the size of an object seen through it, but also resolves its finer details. So this is the background of compound microscope. So basically, compound microscopes helps us in making things bigger. So once the things are bigger, particularly when we are seeing unicellular organisms like amoeba, paramecium, bacteria, so we are able to make it bigger. How much time bigger? In our lab, we have got something like 1000 times we can make it bigger. And the minimum that we can do is 450 times. So 450 times to 1000 times we can make an object bigger so that we can see everything that is inside. So with that, what is the object? To study parts of compound microscope, its proper use and maintenance. Requirement. The requirements are compound microscope. We are having very good compound microscopes, permanent slides, and then practical notebook. And the compound microscope is the most commonly used microscope in a biology laboratory. It magnifies the objects by the combination of two lens systems. One is called objective lens, lies closer to the object, and another is eyepiece or ocular lens, remains close to eye through which you are seeing. The magnifying power of lens is written on it as you have noted, like 5x, 5 times, 10x, 15x, 45x, like this. The magnification of the microscope is the combination of magnification of two lens systems. Thus, magnification of compound microscope is magnifying power of the objective lens. Normally, we are having like 10x. Let's say magnifying power of eyepiece. Eyepiece also 10 into 10. So 100 times we can magnify it. So we have got something like a disc-shaped structure in our compound microscope wherein three lenses are there, like you have got low power, medium power, high power. So you got something like 5x, 10x and 45x. Eyepiece, let's say if we are having 10x, let's say you are keeping this in low power 10 into 10, 100 times. Whereas you are keeping in medium size 15 into 10, 150 times. Whereas you are keeping it 45x, 10 eyepiece, 45 object lens, 45 into 10, 450 times. This is what I said. That is the power of the microscope. So what are the parts of microscope? A compound microscope consists of following parts. All these come under description, mind you. First, foot. The microscope is built around a basal foot. Limb. It is slightly curved part of the microscope which hold the body tube, as you are seeing here. And then third is the stage. You are seeing the stage. Stage is fixed to the limb with the two clips to hold the slide. And the fourth one is mirror. A mobile mirror is fixed at the lowermost part of the limb. Its plane surface is used to direct the light over the object seen under low power, and concave surface is used to converge the light on the object seen under high power. And then diaphragm. And the diaphragm, it is present under the stage to regulate the amount of light entering the microscope. So that controls the light process. And then body tube. It is attached to the upper end. It is attached to the upper end of the limb and can be moved up and down by rack and pinion mechanism. So you can move this body tube up and down. It carries eyepiece at its upper end, as you are seeing. Eyepiece is on the top, and a revolving nose piece, okay, with 2 or 3 objective lenses at the lower end. As I said, in the disc you can see 10x low power objective, 45x high power objective. So this body tube by having the ability to move up and down can magnify the object. And then coarse or fine adjustment. You can see 2 round discs on the handle. So this round, bigger one is called coarse adjustment and the smaller one is called fine adjustment. Coarse and fine adjustment screws are present at the upper end of the limb, which are used to focus with low and high power objective lenses respectively. So, how to use of the microscope? Place the microscope in maximum diffuse light. So, care must be taken that whenever you are placing the microscope, you must place it in light, source of light. Direct sunlight is harmful for the eyes. The northern light is most suitable. If light sources are artificial, filter, preferably blue colored, is used. Adjust the light by turning the mirror towards the source of light and also by moving the condenser. Place the slide on the stage and adjust the object just over the stage aperture. Locate the object and focus it with a low power objective using coarse adjustment. If higher magnification is desired, turn the nose piece to next higher power. So you can always rotate that disc. And when you rotate the disc, how do you know that it is fixed properly? There will be a small sound, tuck sound. So you must hear that sound. Once you hear the tuck sound, that means object lens is fixed. And then use only finer adjustment at this stage. Once you have fixed the object lens, do not use the coarse adjustment. You must use only fine adjustment. Particularly when the object lens you have kept it high power, that is 45X. So because the gap between the object lens and the slide will be very, very narrow. So if you operate coarse adjustment, what happens? The slide will be broken. So take care that slide will not be broken. So what is that you have to use? Fine adjustment only. Always observe with both eyes open. This is the practice that you need to do. Up to class 10th, okay, I have observed you closing both the eyes also. But at least now you must open one eye and slowly open both the eyes. What is the advantage? When both the eyes are open, with one eye you can see through the eyepiece and see the object, so that you are able to see properly. Second eye, you can keep your practical notebook, small 50-page notebook, and draw the diagram. What you are seeing under microscope, you can draw it. That is the purpose why both the eyes should be open. You can also jot down some important points, because there is a next experiment is identification of the objects. Like microbes will be there, then protozoans will be there. All this you have to identify. So while identifying, you need to write certain marks. What are the characteristic features of that? So based on that, you can identify. This identification is a 2 marks question. So there will be one slide will be there in the practical exam, final exam, plus one specimen will be there. So you need to identify. When you identify, you have to write first what is it, on what basis you are telling that that particular object or that particular slide or section is what you are thinking. So there should be reasoning. Care for the microscope. This is very important. Many times you just carry with a single hand. That is not correct. You must handle carefully. One hand you must use it for the arm. Another hand at the base. You must keep support for the base and lift it. Otherwise the mirror will be broken. So be careful while handling it. Before and after use, clean all the lenses and mental parts, including the stage, with a tissue paper. Don't use your handkerchief, for God's sake. Muslin cloth, soft and clean handkerchief. Of course, I advise you not to use your handkerchief. We'll give you tissue paper. Keep the microscope covered when not in use. So keep it inside. Do not remove the object lenses from the nose piece. Do not remove that. Eyepiece only can be removed. Handle the screw, condenser, mirror and stage clips carefully. Care should be taken that objective lens should not touch the slide in any case. And after this, we have got, at the end of the experiment, we have got some Viva, YOC. That means whatever experiment you have done, there will be some questions from the external examiner. You need to answer them. So for this Viva also there is marks. So keeping the Viva is an indicator whether you have understood the questions or not. So below there are 7 questions with answers. Just go through it. So we'll go with the next experiment in the next week. Until then, draw the diagram of the microscope, write all these notes. and keep one separate biology record book. Thank you.